find a link to the notes as well as full transcripts of each lesson. With that, let's start with chapter 1, which is titled The Cell. In this chapter, we'll be covering the following objectives. First, we'll start with
This is your brain when you're struggling to focus. And this is your brain when you're in the zone.
cell theory, which is a set of principles that define what a cell is and how it relates to life itself. These foundational ideas are essential to everything else we'll learn in biology. Next, we'll focus on eukaryotic cells. These are the types of cells that make up plants, animals, fungi, and protists. We're going to look at their internal structures, like their membrane bound organelle, and we're going to discuss how the cytoskeleton helps maintain their shape and organize their contents. We're also going to talk about how eukaryotic cells come together to form tissues. After that, we'll turn our attention to pro. Hello, everyone. My name's Yvonne. Welcome. Hello, everyone. My name's Iman. Welcome back to my YouTube channel. Today, we're going to finish up chapter 1 for our MCAT biology playlist. We made it to objective 3, which is titled Classification and Structure of Prokaryotic Cells. Prokaryotes are the simplest forms of life, and they include all bacteria. Unlike eukaryotic cells, prokaryotic cells do not contain a nucleus or any membrane-bound organelles. Their DNA floats freely in the cytoplasm, and it's typically organized into a single circular chromosome. Now, there are 3 domains that all life can be classified into: bacteria, archaea, and eukarya. Bacteria and archaea contain prokaryotes. Both groups share certain structural similarities, like lacking a nucleus and reproducing through binary fission. But they also differ in important ways. Archaea tend to live in extreme environments, and they have unique features that more closely resemble eukaryotes than bacteria. Bacteria, on the other hand, are more widespread, and they include many species that are either harmless. Hello, everyone. My name's Iman. Welcome back to my YouTube channel. Today, we're going to cover Chapter 2 for our MCAT Biology playlist, and this chapter is titled Reproduction. Here, we're going to cover the following objectives. First, we're going to start with the cell cycle and mitosis. We're going to break this down into 3 parts: the stages of the cell cycle, the molecular control mechanisms that regulate progression through the cell cycle, and then the process of mitosis itself. Then we're going to move into meiosis. This is the specialized form of cell division that produces gametes. Here we're going to talk about how meiosis differs from mitosis, and then we'll explore both meiosis 1 and 2 in detail. Finally, we're going to shift to the reproductive system. We're going to look at both male and female reproductive anatomy. We're going to discuss how sexual development is regulated at the chromosome and hormonal levels. And then we're going to walk through the hormonal patterns and the structural changes that make up the menstrual cycle. With this introduction in place, let's get started with our first objective, which covers the cell cycle and mitosis. In animals, most of the cells in the body are cells, and we describe these cells as being diploid, or 2N. This means that they carry 2 copies of each chromosome, one inherited from each parent. So for every chromosome, there is a homologous partner present. Now, when these cells grow and divide, They follow a carefully regulated process known as the cell cycle. The cell cycle is a series of stages that eukaryotic cells go through to grow, replicate their DNA, and then eventually divide to produce two new cells. For cells that are actively dividing, the cell cycle is divided into four main stages: G1, S, G2, and M. Let's break that down. We're going to start with G1 or GAP1. This is the first step. This is where the cell begins growing and carrying out its normal functions. During this phase, the cell also starts producing more organelles, structures like mitochondria and ribosomes that are necessary for energy production and protein synthesis. Most importantly, there is a checkpoint here where the cell checks that everything is in good shape before moving forward. If conditions are right, the cell enters the S phase or the synthesis phase. This is where DNA replication occurs. Every chromosome makes an exact copy of itself, producing two identical strands called sister chromatids. These sister chromatids stay connected at a central region called the centromere. Next, in G2 or GAP2, the cell continues to grow and it also replicates organelles and other components in preparation for division. Just like in G1, there is another checkpoint here, this time to ensure that DNA replication was successful and that the cell has everything it needs to divide. The final phase is the M phase. This stands for mitosis and cytokinesis. This is where the actual division happens. The nucleus divides first, and then the rest of the cell follows. We're going to go into more details about mitosis in a moment. But these first three stages, G1, S, and G2, they are collectively referred to as interphase. During interphase, the cell is not dividing, but it's very active. It's growing, it's performing its normal functions, and it's preparing for a big event, mitosis. In addition, during this time, the DNA is in a relaxed, uncondensed state known as chromatin, rather than being tightly packed into visible chromosomes. You may also see a label called G0 phase on this diagram. This is a resting state where a cell exits the cycle either temporarily or permanently. In G0, the cell is still alive and performing its normal function, but it's not preparing to divide. Many of the cells in our body, like neurons, spend most of their time in G0. So again, the key phases of the cycle are G1 for growth, S for DNA replication, G2 for final prep, and then M for division. And together, these steps ensure that new cells are made correctly with complete and accurate copies of the genome. Now that we understand the stages of the cell cycle, let's talk about how this entire process is regulated. The cell doesn't just blindly move from one stage to the next. It actually relies on built-in checkpoints to make sure that everything is happening correctly and safely. There are two major checkpoints that we need to know. The first is the G1 to S checkpoint, sometimes referred to as the restriction point. At this checkpoint, what the cell does is it assesses the condition of its DNA. So it checks to see if the DNA is intact and undamaged. And it basically asks, hey, is it safe to replicate? If everything looks good,
Imagine being able to build an app with your name on it just by thinking about it. Meat base 44.
then the cell proceeds into S phase and it begins DNA synthesis. If not, then the cycle is stopped so that the cell can attack repairs. The key protein that enforces this checkpoint is P53. This is often called the guardian of the gene. The second major checkpoint is between G2 and M phase. This checkpoint serves a different purpose. Here, the cell is checking to make sure that it has grown large enough and to make sure that all of the organelles and proteins have been properly duplicated so that it can support division into two daughter cells. Once again, P53 plays a role here by preventing the cell from moving forward if something is off. Now, the follow-up question is, well, how does the cell know when to pass these checkpoints and move into the next phase? This regulation is actually driven by a set of molecules called cyclins and cyclin-dependent kinases, or CDKs. Cyclins are regulatory proteins whose concentrations fluctuate throughout the cell cycle. CDKs are enzymes that are always present in the cell, but they're inactive on their own. In order to work, a CDK must bind a cyclin. When this happens, the CDK becomes activated, and once active, the cyclin CDK complex adds phosphate groups to specific transcription factors. This is called phosphorylation, and then those transcription factors go on to initiate the expression of genes that are required for the next stage of the cycle. So, for example, G1 S cyclins help push the cell from G1 to S phase, while the M cyclins promote entry into mitosis. Now, you can think of it like this: the cyclin is the timer that builds up as the cell gets closer to a checkpoint, and the CDK is the engine. So when the timer hits a certain level, it activates the engine to push the cell forward. Now, here's where things can go wrong. If the checkpoints fail, if, for example, P53 is mutated and it doesn't stop the cell from dividing with damaged DNA, then the cell can continue throughout the cycle and unchecked. And when this happens, damaged cells can divide uncontrollably. And this is a major hallmark of cancer. Many cancers are associated with loss of function mutations in checkpoint proteins like P53. So, to sum up, the cell cycle is tightly controlled by checkpoints and molecular regulators like cyclins and CDKs. But if these controls fail, the consequences can be serious, like cancer. Now, with that, let's take a closer look at mitosis. This is the process that cells use to divide their genetic material evenly between two daughter cells. Mitosis occurs in somatic cells, which are all the cells of the body except sperm and egg. It's used for growth, repair, and general maintenance. The goal is to take one diploid cell, that's a cell with two copies of each chromosome, and produce two identical diploid daughter cells. We're going to walk through each step of mitosis one by one: interphase, prophase, metaphase, anaphase, and telophase. There's also cytokinesis, which is the final physical separation of the cytoplasm. Let's start with interphase, which technically happens before mitosis begins. By this point, the cell has already gone through G1, S, and G2. And remember, during the S phase, the DNA was copied. So instead of having just one of each chromosome, the cell now has two sister chromatids for every chromosome, and they are held together at a region called the centromere. Sister chromatids are genetically identical copies of a chromosome. They're going to be held together after DNA replication, and then they separate during mitosis to ensure that each daughter cell gets a complete set of chromosomes. The centrosomes, which we're going to later discuss, they organize the mitotic spindle. They're also duplicated at this point. Now we enter prophase. For this, I'm going to scroll down to the next page because there's a lot to discuss when it comes to prophase. During prophase, a few major things are happening at the same time to get the cell ready for division. Let's start with the structures that organize the division process, the centrosomes. Each centrosome sits near the nucleus and it is made up of two centrioles. Centrioles are small tube shaped structures that are built from proteins called microtubules. Together, the centrosomes act like command centers that organize the microtubules that are needed for cell division. So, prophase begins. The two centrosomes move to opposite sides of the cell, or what we call opposite poles. Then from each centrosome, long protein filaments called microtubules start growing outwards. These microtubules form the mitotic spindle, which will eventually grab and pull the chromosomes apart. Now, to actually pull the chromosomes, the spindle needs a place to attach. That's where the kinetochores come in. Each duplicated chromosome has a centromere, and sitting on top of the centromere is a protein complex called the kinetochore. You can think of kinetochores as little handles that the spindle fibers grab onto. The specific spindle fibers that connect to the kinetochores are called kinetochore fibers, and they will be responsible for pulling the sister chromatids apart during metaphase. Meanwhile, the chromatid, which is the loose spring-like form of DNA, it begins to condense into visible chromosomes. Each one looks like an X because it consists of two identical sister chromatids. And finally, the nuclear membrane, it starts to break down, giving the spindle fibers access to the chromosomes. So in summary, the centrosomes move apart and build the spindle. The spindle fibers grow out from the centrosomes. The kinetochores on each chromosome act as an attachment point for those fibers, and the nucleus dissolves while the chromosomes condense. All of this prepares the cell for the next stage, which is lining up the chromosomes in the middle during metaphase. In metaphase, the chromosomes align along the metaphase plate, which is an imaginary line that runs down the center of the cell. Each chromosome is attached to spindle fibers coming from both sides of the cell via its kinetochore. So this symmetrical setup really ensures that each daughter cell will receive one complete copy of each chromosome. Then we move into anaphase. So during this stage, the sister chromatids are being pulled apart at the centromere. Each chromatid is now considered an individual chromosome, and they're dragged towards opposite poles of the cell by shortening of the spindle fibers. Now you'll notice that the chromosome count temporarily doubles here. For example, what was two chromosomes becomes four single chromosomes. But remember, that's just because we're separating duplicates. After that, we enter telophase. At this point, a new nuclear envelope begins to form around each set of chromosomes on either side, and the chromosomes begin to decondense back into chromatid. And then the spindle apparatus also breaks down. Finally, we reach cytokinesis, which is not technically a part of mitosis, but it happens alongside telophase. This is the actual splitting of the cytoplasm and organelles, and it results in two separate daughter cells, each with its own complete nucleus and set of organelles. Both of these cells are diploid, just like the original cell. So that's mitosis for us. Let's go ahead and now move into our third objective, where we're going to discuss meiosis. So unlike mitosis, which happens in somatic or body cells, Meiosis only occurs in gametes, that is, cells that will become sperm or eggs. And the purpose of meiosis is different too. So instead of making two identical diploid cells, meiosis produces four non-identical haploid cells, each with half the number of chromosomes as the original. Let's define that quickly. So a haploid cell has only one copy of each chromosome. So in humans, that's 23 total chromosomes. A diploid cell, on the other hand, has 46 copies of each chromosome. Now, the reduction in chromosome number is really essential for sexual reproduction, so that when sperm and egg unite, the resulting zygote has the correct diploid number. Now, meiosis happens in two rounds, meiosis 1 and meiosis 2. And we're going to go step by step through both. Let's start at the top of the diagram with interphase. Again, this happens before meiosis begins, just like it does before mitosis. And during interphase, the DNA is copied. So each chromosome now consists of two sister chromatids that are joined at the centromere. Then we begin meiosis 1, which is sometimes called the reductional division because it reduces the chromosome number from diploid to haploid. The first phase of meiosis 1 is prophase 1. So here again, we're going to scroll down and take a moment to look more closely at what happens in prophase 1. Prophase 1 is one of the most important stages in all of meiosis because this is where we introduce genetic variation into the next generation. First, homologous chromosomes find each other and pair up. Homologous chromosomes are the two versions of a chromosome, one from your mommy, mamacita, and one from your father. And they carry the same kinds of genes, just possibly different versions. And when they pair up side by side, we call the structure they form a tetrad because the same one. The first, which is sometimes called the reductional division because it reduces the chromosome number from diploid to haploid. The first phase of meiosis 1 is prophase 1. So here again, we're going to scroll down and take a moment to look more closely at what happens in prophase 1. Prophase 1 is one of the most important stages in all of meiosis because this is where we introduce genetic variation into the next generation. First, homologous chromosomes find each other and pair up. Homologous chromosomes are the two versions of a chromosome, one from your mommy, mamacita, and one from your father. And they carry the same kinds of genes, just possibly different versions. And when they pair up side by side, we call the structure they form a tetrad because it includes four chromatids total, two from each chromosome. This pairing process is called synapsis. Now, once they're paired, the chromosomes can actually exchange pieces of DNA. The exchange happens at a point called the chiasmata, and the process as a whole is called crossing over. And it allows segments from the maternal and paternal chromosomes to swap. The result is that we no longer have exact copies of mom and dad's chromosomes. We now have recombinant chromosomes. These are hybrids that contain a mix of genetic material from both parents. This process increases genetic diversity in offspring. Finally, this genetic recombination also helps to unlink genes that are usually inherited together, which gives us even more variety. So to summarize prophase 1, homologous chromosomes pair up through synapsis, crossing over occurs at the chiasmata, and the result is recombinant chromosomes that promote genetic diversity. Now let's return to our main diagram and continue with meiosis 1. After prophase 1, we move into metaphase 1. Here, the homologous chromosomes pairs and they line up at the metaphase link. Importantly, they line up in a random orientation, which adds even more genetic variation. So one chromosome from each pair is facing each pole of the cell. Then comes anaphase 1. Unlike mitosis, where sister chromatids are pulled apart, here in meiosis 1, it's the homologous chromosomes that are separated and pulled to opposite poles. The sister chromatids stay together for now. Next is telophase 1 and cytokinesis. So here a new nuclear membrane may start to form around each group of chromosomes, and the cell physically splits into two daughter cells. Each one is now haploid. It contains just one chromosome from each original pair, even though each chromosome is still made of two sister chromatids. Now we move into the second part of meiosis called meiosis 2. This round is a lot like mitosis, and it's called the equational division because the chromosome number stays the same. It's still haploid before and after, but the sister chromatids are finally separated. So we begin with prophase 2, where the spindle forms again in each of the two haploid cells. Then in metaphase 2, the chromosomes line up at the center of each cell. This time it's individual chromosomes, not pairs. In anaphase 2, the sister chromatids are finally pulled apart and dragged towards opposite poles. And then finally, in telophase 2 and cytokinesis, the nuclear membranes reform and both cells divide once more. The result is 4 haploid daughter cells, each genetically unique. In sperm cells, all 4 of these will become functional sperm. In females, only one becomes a mature ovum, but we're going to talk about that later. One side note that I want to make here before we move on is about non-disjunction. This is when chromosomes fail to separate properly during meiosis, and it can happen either in anaphase 1 or in anaphase 2. If it happens in anaphase 1, the homologous chromosomes don't separate. That means one cell ends up with both chromosomes from the pair, and the other gets none. If it happens in anaphase 2, the sister chromatids don't separate. So again, one resulting cell ends up with an extra chromatid, and one gets none. Either way, the result is gametes with an abnormal number of chromosomes, either too many or too few. If such a gamete is involved in fertilization, only one becomes a mature ovum, but we're going to talk about that later. One side note that I want to make here before we move on is about non-disjunction. This is when chromosomes fail to separate properly during meiosis, and it can happen either in anaphase 1 or in anaphase 2. If it happens in anaphase 1, the homologous chromosomes don't separate. That means one cell ends up with both chromosomes from the pair, and the other gets none. If it happens in anaphase 2, the sister chromatids don't separate. So again, one resulting cell ends up with an extra chromatid, and one gets none. Either way, the result is gametes with an abnormal number of chromosomes, either too many or too few. If such a gamete is involved in fertilization, then the resulting zygote will also have the wrong number of chromosomes. And this can lead to genetic conditions like Down syndrome, which is caused by an extra copy of chromosome 21. So while meiosis is usually very precise, even small errors like non-disjunction can have major consequences in development. So the big picture is the following. Meiosis 1 separates homologous chromosomes and it reduces the chromosome number. Meiosis 2 separates sister chromatids and it maintains chromosome number. Together, they produce 4 genetically diverse haploid cells. Now, let's take a moment to compare mitosis and meiosis side by side. This might be repetitive, but I really want to make sure we understand mitosis and meiosis really well. These two processes can feel similar, especially because they share many of the same names for these stages, but their outcomes and their purposes are fundamentally different. Let's start with this. In mitosis, a diploid cell, written as 2N, divides to form two identical diploid cells. So the chromosome number stays the same from start to finish. In meiosis, a diploid cell undergoes 2 rounds of division to produce 4 haploid cells, each with half the original number of chromosomes. So here we go from 2N to N. Now let's talk about where these processes happen. Mitosis occurs in all dividing cells of the body. Your skin cells, your blood cells, liver cells, you name it. Meiosis, on the other hand, it occurs only in your germline cells that give rise to sperm or egg. It's a specialized process for sexual reproduction. Another big distinction is how the chromosomes behave. In mitosis, homologous chromosomes do not pair up. The chromosomes line up independently, and it's the sister chromatids that get pulled apart. But in meiosis, specifically meiosis 1, the homologous chromosomes pair up and then they are separated. And this pairing is what allows for crossing over to occur, which brings us to another key difference. Crossing over, or this exchange of genetic material between homologous chromosomes, it never happens in mitosis. But in meiosis, particularly during prophase 1, crossing over is a defining event. It introduces genetic variation and it ensures that no two gametes are exactly alike. So to summarize, mitosis makes identical cells for growth and repair. Meiosis makes unique sex cells for reproduction. They're both essential, but they serve completely different roles. And with that, we can move into our third and final objective, titled The Reproductive System. We'll start with a look at chromosomal sex determination. Humans have 23 pairs of chromosomes, and it's the 23rd pair, known as sex chromosomes, that determine biological sex. If the individual inherits 2X chromosomes, they're going to develop as genetically female. If they inherit one X chromosome and one Y chromosome, then they're going to develop genetically male. Let's look closely at the X and Y chromosomes because their structure and function play a very key role in inheritance patterns. The X chromosome is large and it carries hundreds of genes. A lot of them are actually unrelated to sex. But because of its size and gene density, mutations in genes on the X chromosome can lead to things like X-linked genetic disorders. The Y chromosome, by contrast, is much smaller. Now, some may say it's average, but nevertheless, it contains far fewer genes. Its most critical gene is the SRY gene, which stands for sex determining region Y. And this gene triggers the development of testes during embryogenesis, which in turn leads to the production of testosterone and the development of male reproductive structures. Now, I want to focus really quickly on the genetics of X linked traits. In females, which have XX, each gene on the X chromosome it exists in two copies, one on each X. So for any given gene, a female can be homozygous dominant, where both alleles are functional. It can be homozygous recessive, where both copies carry a mutation, or it can be heterozygous, where one allele is normal and the other carries a mutation. So, for any given gene, a female can be homozygous dominant, where both alleles are functional. It can be homozygous recessive, where both copies carry a mutation, or it can be heterozygous, where one allele is normal and the other carries a mutation. Homozygous recessive, where both copies carry a mutation, or it can be heterozygous, where one allele is normal and the other carries a mutation. Now, in the heterozygous case, it can be homozygous recessive, where both copies carry a mutation, or it can be heterozygous, where one allele is normal and the other carries a mutation. Now, in the heterozygous case, the dominant functional allele is often sufficient to compensate for the mutated one. So these individuals are referred to as carriers. They typically do not express the disorder themselves, but they can pass the mutated allele on to their offspring. In males, however, they have XY and only one X chromosome. That means they are hemizygous for X linked genes. Males, however, they have Y and only one X chromosome. That means they are hemizygous for X linked genes, and they don't have a second copy to compensate. So if a male inherits a mutated X linked gene, then whether it's dominant or recessive, the mutation is going to be expressed because there's no corresponding allele on the Y chromosome to mask its effect. This is super important. I'm going to repeat. So if a male inherits a mutated X-linked allele, regardless of whether it's dominant or recessive, the mutation will be expressed. This is why X-linked recessive disorders are more common and more severe in males than in females. So to summarize this part, females XX have two alleles for each X-linked gene, and they can be homozygous or heterozygous. Heterozygous females may be carriers of X-linked recessive disorders. Males, which are XY, are hemizygous, so they will express any mutation that is present on their single X chromosome. Now that we've covered that, let's turn our attention to the male reproductive anatomy. You can see in the diagram here that the male reproductive system consists of internal and external structures. Here we're going to go over how everything works, and then in the second page, we will reiterate using a table that summarizes everything. We're going to start from where sperm is made and then work our way up to ejaculation. So the testes is where we're going to start. These are housed in the scrotum, and they have two main jobs: producing sperm and secreting testosterone. Inside the testes are the seminiferous tubules. These are long, coiled structures where sperm production actually occurs. Within these tubules, we have Sertoli cells. These provide structural and nutritional support to developing sperm. And we also have the Leydig cells, which are located outside the tubules. They secrete testosterone and other androgens, which are essential for male secondary sex characteristics and also for maintaining sperm production. Now, the scrotum, it hangs outside the body and it helps regulate temperature. Sperm development really requires a temperature that's slightly lower than the core body temperature, and the scrotum achieves this by contracting or relaxing in response to heat. Once sperm are made, they move into the epididymis. This is a long coiled tube where they mature and where they are stored. It's here that sperm gain motility, which is their ability to swim. Then during ejaculation, sperm travels through the vas deferens. This is a muscular tube that propels sperm upward towards the urethra. It also joins with the ejaculatory duct, which merges with secretions from various accessory glands to form semen. Those accessory glands are also important to know. So we'll start with the seminal vesicle. This is an accessory gland that produces a fluid rich in fructose, which provides energy for the sperm. We also have the prostate gland, where they mature and where they are stored. It's here that sperm gain motility, which is their ability to swim. Then during ejaculation, sperm travels through the vas deferens. This is a muscular tube that propels sperm upward towards the urethra. It also joins with the ejaculatory duct, which merges with secretions from various accessory glands to form semen. Those accessory glands are also important to know. So we'll start with the seminal vesicle. This is an accessory gland that produces a fluid rich in fructose, which provides energy for the sperm. We also have the prostate gland, which contributes a slightly alkaline fluid that helps sperm survive in the acidic vaginal environment. There's also the bulbourethral gland, also known as Cowper's gland. This secretes a clear, lubricating mucus that helps flush out residual urine from the urethra, and it also prepares it for the passage of semen. Finally, semen exits the body through the
My wife stopped trusting me because I never do the things I say I'll do. Just does everything herself now.
through the urethra, which runs through the penis. The penis is the external organ that's responsible for delivering sperm to the female reproductive tract. Now, here in this table, you'll see all of those parts we just discussed and their main function summarized. So I'm going to reiterate. Testes, they produce sperm, they secrete testosterone. The seminiferous tubules, they are the site of sperm production. For delivering sperm to the female reproductive tract. Now, here in this table, you'll see all of those parts we just discussed and their main function summarized. So I'm going to reiterate. Testes, they produce sperm, they secrete testosterone. The seminiferous tubules, they are the site of sperm production. The Sertoli cells nourish developing sperm cells. The Leydig cells secrete testosterone and other androgens. The scrotum helps maintain proper temperature of the testes. The epididymis stores sperm and it allows them to gain motility. The vas deferens transports sperm from the epididymis to the ejaculatory duct. The ejaculatory duct merges with the vas deferens and it leads into the urethra. The urethra carries semen and urine through the penis and out of the body. The seminal vesicles produce fluid to nourish the sperm. The bulbourethral gland secretes clear fluid to help clean out urine. and it also lubricates the urethra during arousal. The prostate gland produces fluid to protect sperm and vagina, and then the penis is the organ that's used to deliver semen. From here, it's important for us to learn about spermatogenesis and the structure of a mature sperm. Spermatogenesis is the formation of sperm cells through meiosis. This process starts at puberty and it continues throughout a male's life. Here's the step-by-step sequence. It all begins with spermatogonium. This is a diploid stem cell that undergoes mitosis to replenish the stem cell pool and produce cells that are ready to enter meiosis. One of those daughter cells differentiates into a primary spermatocyte, which is still diploid. The primary spermatocyte enters meiosis 1 and it divides into two secondary spermatocytes, which are haploid. Each secondary spermatocyte undergoes meiosis 2, producing two spermatids, so we now have four haploid cells. These spermatids then go through a process called spermiogenesis, where they mature into spermatozoa, fully formed sperm with a head, midpiece, and tail. On that note, we want to elaborate on those sperm structures. The head contains the nucleus, which carries the father's genetic material. At the very front is the acrosome. This is a cap-like structure filled with enzymes that help the sperm penetrate the egg during fertilization. The midpiece is packed with mitochondria that produce ATP to fuel the sperm's movement. And then the tail is a whip-like structure that propels the sperm forward, allowing it to swim through the female reproductive tract. On that note, let's now shift our focus to the female reproductive system. Unlike the male reproductive system, which is primarily external, female reproductive organs are almost entirely internal. These organs are responsible for producing ova, the female gametes, as well as supporting fertilization, implantation, and fetal development. Let's start with the gonads. In females, the gonads are the ovaries. These are paired oval-shaped organs that are located on either side of the uterus, and they serve two key functions. The first is to produce and release ova, which is the eggs, and the second is to synthesize and secrete the female sex hormones, estrogen and progesterone. Each ovary contains thousands of small fluid-filled sacs that are called follicles. And every follicle houses an immature oocyte or egg cell, and it provides protection and nutrients as the egg develops. Now, once per menstrual cycle, in response to hormonal signals, one follicle becomes dominant and it will undergo ovulation. This is the process in which the mature egg is released from the ovary. More importantly, ovulation doesn't release the egg directly into the fallopian tube. It first enters the peritoneal cavity, which is the fluid-filled space that surrounds the abdominal cord. From there, the egg is drawn into the fallopian tube, also known as the uterine tube. The part of the tube that captures the egg is called the infundibulum, and it is lined with fimbriae. These are finger-like projections that will gently sweep the egg in. Now, the fallopian tube is the typical site of fertilization, where a sperm cell may encounter and penetrate the egg. And if that fertilization occurs, then the zygote will continue moving through the part of the tube that captures the egg is called the infundibulum, and it is lined with fimbriae. These are finger like projections that will gently sweep the egg inward. Now, the fallopian tube is the typical site of fertilization where a sperm cell may encounter and penetrate the egg. And if that fertilization occurs, then the zygote will continue moving through the tube towards the uterus. The uterus is a strong muscular organ, and its main role is to support and nourish a developing embryo. The inner lining of this uterus is called the endometrium, and it thickens each cycle in preparation for potential implantation. But if no fertilization occurs, then this lining is what's shed during menstruation. At the lower end of the uterus is the cervix. This serves as the gateway between the uterus and the vaginal canal. Now, during most of the cycle, the cervix remains closed, but during ovulation, it softens and it opens slightly to allow sperm entry. During childbirth, also, the cervix will dilate a lot, significantly, to, of course, allow the passage of the baby. The vagina is the muscular tube that extends from the cervix to the external opening of the body. And it serves multiple functions, two of which are the passageway for menstrual flow, and of course, the birth canal during delivery. Finally, as for the external genitalia, this is collectively referred to as the vulva, and it includes the labia majora and the labia minora, which are folds of skin that protect the vaginal opening. We also have the greater vestibular glands, which secrete a lubricating fluid that will help reduce friction during sexual intercourse. And then there is the hymen. This is a thin membrane that may partially cover.
Oh, jeez, finally 10 minutes of rest. Oh my God. Uh, I just wanna, I don't even know. I don't even know what I'm doing here, honestly. Was the first 50 minutes, you guys. I couldn't even read or do anything because I knew I would get, distracted pretty easy. And it's so good to be living right now because I'm not touching my phone. That was the main goal, not touching my phone. Because even when I hide it, I always find a way to like reach for it, whatever I'm gonna break. So now that I'm using it, I don't have any choices. Like it has to stay there. Oh my goodness. It was dang, studying can be painful, you guys. I'm really trying to romanticize it. It's really not something that I like. Now I'm doing biology. I'm on the chapter about the reproductive system, as you guys may have heard. Um, you know, talking about XX, XY, yeah, cell cycle, mitosis, meiosis. Um, yeah, it's good. It's a good review. It's a good review. It's always good to train your brain every single day. It's because I'm also supposed to be mastering it now. Um, so yeah, I'm trying. Okay, I'm trying my best right now. But yeah, like, dang, I don't even know. Like sitting down here is just so humbling. It's so humbling. Ah, we have like about what 10? No, 5 more hours of studying. I'll see how I feel because I'm also doing my laundry. Um, but yeah, my goal is to reach at least 3 hours right now. If I want to take a 2 hours break, why not? And then coming back again to the 3 hours left. But honestly, I'm I'm kind of shocking myself right now. This is my last hope. Honestly, me doing that is really just me reaching my last hope. I'm tired. I'm just so ah I'm just feeling too much right now. Too many things going on and I'm overwhelmed, honestly, with everything happening right now. With like studying and things in general, like even when it comes to school, I'm very overwhelmed and I'm really trying to like prioritize what I need to prioritize right now because it's really easy to just get overwhelmed and skipping every single thing that you're supposed to be doing because while you feeling bad, your school grades and everything are not even waiting for you. If the professor says tomorrow you're having an exam, you're gonna have the exam. It's not because you feel bad today that you're gonna have the grace to skip it. You know what I mean? So I'm really trying to force myself and learn to just keep moving even though when I really don't feel like it. Some people gonna say it's just, discipline. You cannot be motivated every single day. But I've reached a point where even discipline is hard for me. Like I wake up in the morning and I don't even want to wake up because I'm essentially not like I don't feel like I have a purpose. Not to that point, okay? I'm just very my battery is at -1 right now. If that makes sense. It doesn't look like it, but I'm really trying to survive because it's a part of life. Not every single day you're gonna feel great. Maybe tomorrow I'm gonna feel the best, and then it just happens. But it is one of these days where I'm feeling really, at my lowest, and I'm really trying to still do my best because when exams are going to come and everything, I'm gonna feel even worse because that whole time I was just ranting and you know not even trying to do something that will help me out whenever I have to be responsible and do what I need to do. So yeah, I don't want to self sabotage as well because I tend to do that. I generally tend to do that. Um, yeah, 5 more minutes, you guys. I'll be there for a long time, so you can even put it somewhere, sit down if you want to study a little bit. If the music or the video I'm watching is bothering you, you can even like mute the thing and put your own music and having the live in the background, It's not that bad. Yeah. Actually my second time drinking water since this morning. It's already 1:30. And I'm wondering why I have an upset stomach. I'm not even drinking enough water. That's bad. It's just they're drinking coffee. Um, it's a cafe latte, high protein coffee. It's 30 gram of protein. I got it from Costco. Very good. Not gonna lie, very good. I'm supposed to um drink it with some ice and all that thing, but I just get the box and I drink it like that because I'm tired. I'm tired, okay? I'm tired. I don't even want to make any effort. When it comes to eating and everything, I'm like, heck no. Matter of fact, somebody says studying for an exam tomorrow. I procrastinated for a long time. My action are caught up to me now. That's exactly what I was talking about. Like procrastinating or self-sabotaging, just feeling bad in general and you want to push everything, to the next day until the day actually comes and now you're rushing, stressing out and everything. That's what I need to avoid. No, not gonna lie, right now I'm kind of cooked, but I'm trying to stay optimistic and be like, it's fine, I'm gonna figure that out. I'm gonna figure that out. It's better than nothing, actually. It's better than nothing. I wish I was strong enough to just take every single day to discipline, but sometimes it's just like that where I don't even know how to feel. I'm just doing stuff. There is no purpose. I don't even feel like doing anything, like, honestly. Oh yeah. What you studying? I'm studying biology. It's basically um MCAT biology review situation from Professor Iman. She's on YouTube. Very, very good. She has a whole playlist for organic chemistry, physics, biology, uh, biochemistry, and I believe that's all. She doesn't have behavioral science like everything that is sociology or psychology, but it's fine. I feel like sociology and psychology is more like you can watch other videos. Like there are other people who go through it pretty well, or just Khan Academy, if I'm not mistaken. They also have good videos about that. But behavioral science is this section is kind of like a new, a whole new language for me. So I was just, I would just expose myself directly to it and try to figure out what you're talking about. So basically, doing some question samples, that would be great, because honestly, sitting down and learning all that, Hey girl, I just joined. How are you and how is the study going? Hi. Um, it's going. It's going. I'm doing 50 minutes in, 10 minutes out Pomodoro method. That's why you have all the music going on. It helps me out. Otherwise I get very overwhelmed. I feel like I should have picked maybe a 30-minute situation or a 25-minute, but I feel like I would have been doing too many breaks. That's why. But yeah, that's what I'm doing now. It's my first hour, about to finish in 1 minute exactly, and then we're gonna keep going. I'm supposed to be here for 6 hours, but we'll see how it goes. Okay, we'll see how it goes. I've been really lacking on my studying, so me deciding to sitting down for 6 hours might be too ambitious. But I'm not losing anything by doing that, so I don't mind. And this is what I'm supposed to be doing, so I'm gonna just keep pushing. 30 more seconds and I'll be there. If you guys want to you know, uh write some stuff and whatever, please. No worries. If I'm not watching, while I'm doing this, it's fine. It's fine. I'm really trying to focus in on my break. I'll be right back to read and respond. Margaret says it's going. Don't worry. You got this, babe. Thank you. Thank you. Oh my goodness. Right now, also studying. I have cash obsession, a kind of second chancing. Oh, nice. Oh, girl, we got this. Okay, let's go back again. Talking about the women reproductive system. Let's go.
The vaginal opening in some individuals. And of course, the vaginal opening, this is the external exit of the reproductive tract. So, to summarize, the ovaries produce eggs and ovum. The fallopian tubes capture the egg and they serve as the site of fertilization. The uterus supports the pregnancy. The cervix controls access between the uterus and the vagina. And the vagina serves reproductive, menstrual, and birthing functions. With that, let's now turn our attention to the production of female gametes. This is a process known as oogenesis, and oogenesis refers to the development of a mature egg cell or ova from an original stem cell. Just like spermatogenesis in males, this process involves meiosis, but with some important differences in timing and outcome. So let's walk through the developmental sequence step by step. The process begins with a diploid stem cell called the oogonia. All the oogonia a female will ever have are formed during fetal development. This is unlike males who produce new sperm throughout their life. These oogonia, they undergo mitosis to multiply, and then they begin meiosis. Once they enter meiosis 1, they are called primary oocytes. However, these cells pause in prophase 1 and they remain in that arrested state until puberty. So when a female is born, she already has all of her primary oocytes sitting in her ovaries, pretty much frozen mid meiosis. Then, starting at puberty, each menstrual cycle, there are hormonal signals that stimulate one primary oocyte to continue meiosis. It completes meiosis 1 and it forms two cells, a large cell called the secondary oocyte and a small non-functional cell called the first polar body. The polar body is essentially a way to discard extra chromosomes while still preserving most of the cytoplasm in the secondary oocyte. The secondary oocyte then begins meiosis 2, but again, It pauses, this time in metaphase 2. It will not complete meiosis unless fertilization occurs. If the sperm penetrates the secondary oocyte, then meiosis 2 finishes and it produces the mature ovum and another small polar body, which again discards excess genetic material. By the way, I really want to emphasize this, polar bodies are small cells produced during each meiotic division, and they contain a nucleus and chromosomes, but very little cytoplasm. They are not functional yet. Their sole purpose is to eliminate the extra sets of chromosomes during the reduction process. Only one functional egg is produced per meiosis, unlike males, where meiosis yields 4 functional sperm. Now, another structural feature to note is that the oocytes are surrounded by 2 protective layers. You have the zona pellucida. This is a thick glycoprotein rich layer that lies directly around the plasma membrane of the oocyte, and it plays a key role in sperm binding and initiating the acrosome reaction during fertilization. Then we have the corona radiata, which is a layer of granulosa cells that surround the zona pellucida. These cells are derived from the follicle, and they're important for nourishment and communication between the follicle and the egg. With that, we want to follow this
This is Pocket. I never go anywhere without it. At the tap of a button, it will
discussion on how everything is hormonally regulated in both males and females. At the top of this cascade is the hypothalamus. This is a region of the brain that functions as the body's master regulator. And it releases a hormone called gonadotropin releasing hormone. This hormone acts on the anterior pituitary gland. is the hypothalamus. This is a region of the brain that functions as the body's master regulator. And it releases a hormone called gonadotropin releasing hormone. This hormone acts on the anterior pituitary gland and it essentially promotes the release of two very important signaling molecules: follicle stimulating hormone, FSH, and luteinizing hormone, LH. These two are collectively called gonadotropins because they target the gonads, the testes in males, and the ovaries in females. But the way they act depends entirely on the sex specific tissues that are present. So let's start with discussing what FSH and LH do in males. Follicle stimulating hormones in males act on the Sertoli cells inside the seminiferous tubules of the testes. In males are present. So let's start with discussing what FSH and LH do in males. Follicle stimulating hormones in males act on the Sertoli cells inside the seminiferous tubules of the testes. These Sertoli cells are crucial for supporting and nourishing developing sperm. So essentially, FSH is the hormone that drives spermatogenesis, which is the process of producing sperm cells. Without it, the entire internal environment that is required for sperm development would absolutely collapse. What about luteinizing hormone? It acts on a different type of cell known as the Leydig cells, which are located in the spaces between these seminiferous tubules. In response to luteinizing hormone, these Leydig cells begin producing testosterone. And by the way, testosterone is the primary male sex hormone. Testosterone is not only essential for the continued support of spermatogenesis, but it's also really important for the development of male secondary sexual characteristics. And this includes things like the deepening of the voice, growth of facial and body hair, increased muscle mass, and even the enlargement of the penis and testes during puberty. It also contributes to libido and just overall male reproductive health. Now let's turn to females. In females, FSH plays a really central role in the development of ovarian follicles, which are small sacs in the ovaries that house and nourish immature oocytes. Early in the menstrual cycle, a group of follicles begin to grow under the influence of FSH, but typically only one actually becomes the dominant follicle that matures fully. And as this follicle develops, it starts producing increased amounts of estradiol, which is a potent form of estrogen. As for luteinizing hormone, this is the hormone that triggers ovulation, which is the release of the oocyte from the dominant follicle. This happens around day 14 of the menstrual cycle, and it's driven by a surge in luteinizing hormone levels. That LH surge is actually caused by high levels of estradiol that's exerting a temporary positive feedback effect on the hypothalamus and the pituitary. And that pushes luteinizing hormone secretion up. Once ovulation occurs, though, the empty follicle transforms into a structure that is called corpus luteum, which begins producing progesterone along with some estrogen. And all of the points we just discussed are written here as a couple of elements. Now that we've talked about hormonal regulation, let's walk through the menstrual cycle itself. This cycle usually lasts around 28 days and it's divided into four phases: the follicular phase, ovulation, luteal phase, and menstruation. Each phase is defined by a specific hormonal profile and coordinated changes in both the ovaries and the uterus. We're going to start with the follicular phase, which begins on day 1 of the cycle. This is the same day that menstruation starts. At this point, levels of estrogen and progesterone are low because the corpus luteum from the previous cycle has broken down. And this drop in hormone lifts the inhibition on the hypothalamus and the pituitary, which leads to the release of gonadotropin release hormone. This is going to stimulate the anterior pituitary release, lifts the from the previous cycle has broken down. And this drop in hormone lifts the inhibition on the hypothalamus and the pituitary, which leads to the release of gonadotropin releasing hormone. This is going to stimulate the anterior pituitary to release follicle stimulating hormone and luteinizing hormone. FSH causes a group of ovarian follicles to then begin maturing, and usually one becomes dominant. As it grows, that dominant follicle secretes the increasing amount of estradiol, which is a potent form of estrogen. Estradiol thickens the endometrial lining and it prepares the uterus for potential implantation. Next is ovulation, which typically occurs around day 14. Here, estradiol levels rise, and once they stay high long enough, the feedback switches from negative to positive, and that triggers a sharp increase in LH secretion. This is known as the LH surge. That LH surge causes the dominant follicle to rupture and release a secondary oocyte, and this oocyte is swept into the fallopian tube where it might encounter sperm and become fertilized. Now, ovulation, it marks the end of the follicular phase and the start of the luteal phase. Now, we enter the luteal phase. Again, this comes right after ovulation. Here, the ruptured follicle turns into the corpus luteum, which begins secreting progesterone during the luteal phase. Now, we enter the luteal phase. Again, this comes right after ovulation. Here, the ruptured follicle turns into the corpus luteum, which begins secreting progesterone and some estrogen. Progesterone is the dominant hormone in this phase. It stabilizes and it maintains the endometrial lining. At the same time, progesterone is going to suppress gonadotropin releasing hormones, FSH, and LH to prevent the start of a new cycle. And this phase lasts about 14 months. Gonadotropin releasing hormones, FSH, and is the endometrial lining. At the same time, progesterone is going to suppress gonadotropin releasing hormones, FSH, and LH to prevent the start of a new cycle. And this phase lasts about 14 days. If no fertilization occurs, the corpus luteum breaks down into the corpus albicans, and hormone levels are fall off. This leads into the final phase, which is menstruation. The drop in estrogen and progesterone is going to cause the endometrial lining to break down and be shed. And this is what we see as our periods that come every month. Menstruation is typically going to last 3 to 7 days, and it marks the beginning of the next cycle. But if fertilization does occur and the embryo implants successfully, the embryo's trophoblast cells begin producing human hormone. Fertilization does occur at the beginning of the next cycle. But if fertilization does occur and the embryo implants successfully, the embryo's trophoblast cells begin producing human chorionic gonadotropin, or HCG. HCG, chorionic gonadose trophoblast cells begin. Producing human chorionic gonadotropin, or HCG. HCG maintains the corpus luteum, which continues to produce progesterone and estrogen, and that keeps the uterine lining intact and it supports early pregnancy. HCG is also the hormone detected by pregnancy tests. Actually, as a person ages, the cycle becomes less regular. That's because the number of follicles declines and the ovaries become less responsive to FSH and LH. Ovulation then becomes less frequent and hormone production decreases. And this transition is called menopause. It usually occurs between the age of of 45 and 55, and it's marked by the permanent end of menstruation, a lasting drop in estrogen and as a result, a lot of people experience symptoms like hot flashes. Mood changes, sleep disturbances, sometimes bone density loss. Well, so again, the menstrual cycle includes the follicular phase, ovulation, the luteal phase, and menstruation. And each step is driven by hormonal feedback loops between the brain, ovaries, and uterus. And together they keep the cycle moving in a highly coordinated way. With that, we've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck. Happy study.
Something, if something's great, people just can't resist it. It doesn't matter what it is. It's the picture pretty.
Hello, everyone. My name's Iman. Welcome back to my YouTube channel. Today, we're going to tackle a practice problem set that relates to our lecture on research. Let's go ahead and get started with problem number one. Problem number one says, which of the following is the correct sequence of the development of a mature sperm cell? In our lecture, we covered spermatogenesis. This is the process of sperm production in males. We always begin with a spermatogonium. This is a diploid stem cell, and it divides by mitosis to either replenish the stem cell population or give rise to the next stage. From there, it becomes a primary spermatocyte, which is still diploid, and it's about to enter meiosis 1. After meiosis 1, each primary spermatocyte gives rise to two secondary spermatocytes, which are haploid but still have duplicated chromosomes. These secondary spermatocytes then quickly enter meiosis 2 and they produce a total of four spermatids. Finally, spermatids undergo a process called spermiogenesis where they mature into spermatozoa or mature sperm cells. So, putting this all together, the correct developmental sequence matches answer choice B. One is B. Let's go ahead and move into problem 2. Problem 2 says, which of the following correctly pairs the stage of development of an egg cell with the relevant point in the life cycle? First, remember that in females, all of the primary oocytes are actually formed before birth. They begin meiosis 1 while the individual is still in utero, but then they pause in prophase 1. That means from birth all the way through childhood and even up to puberty, all the oocytes are sitting in prophase 1. They don't assume meiosis until ovulation begins during puberty. Now, during each menstrual cycle, one oocyte completes meiosis 1 and enters meiosis 2. But here's the key detail, it doesn't complete meiosis 2 unless fertilization happens. Instead, it gets arrested at metaphase 2 and it stays there until a sperm arrives. So, the moment of ovulation, the secondary oocyte is released and it is stuck in metaphase 2. It will only finish meiosis 2 if it's fertilized. Keeping that all in mind, the correct answer here is going to be C. At ovulation, the oocyte is in metaphase 2. Problem 3 says some studies suggest that in patients who have Alzheimer's disease, there is a defect in the way that the spindle apparatus attaches to the kinetochore fibers. At which stage of mitotic division would one first expect to be able to visualize this problem? Let's go through the phases. In prophase, the chromosomes condense and the mitotic spindle begins to form. This is also the stage when these spindle fibers start attaching to the kinetochores, which are protein structures at the centromeres of the chromosomes. So if there's a problem with that attachment, this is the first time you'd actually be able to observe it. because this is the first time the process is happening. In fact, A is the correct answer, but we're still going to work through the rest of the process. In metaphase, the chromosomes line up at the metaphase plate, but by this point, the spindle should have already attached. If it didn't, you'd see something off, but the problem itself began in prophase. Anaphase is when these sister chromatids separate and move to opposite poles. So if attachment failed earlier, this stage would just reflect the consequences. And telophase is when the chromosomes decondense and the nuclear envelope reforms, so it's already too late to notice the attachment issue. So again, the question is asking when you'd first be able to notice the issue, and the answer to that is going to be prophase. That's when spindle fibers start reaching out to connect the kinetochores. So that's when you see if something isn't working properly. 3 is A. Problem 4 says a researcher wishes to incorporate a radiolabeled deoxyadenine into the genome of one of the two daughter cells that would arise as a result of mitosis. What is the latest stage of cellular development during which the radio-labeled deoxyadenine could be added to achieve this result? The key here is understanding when DNA is actually being synthesized, because that's when nucleotides like deoxyadenine can get added into the DNA strand. DNA replication happens during the S phase, or the synthesis phase of the cell cycle. This is the only time that the cell is copying its DNA in preparation for division. So if the goal is to get that radio-labeled nucleotide into the DNA, it has to be added while replication is happening. So during the S phase. If you wait until G2, the DNA has already been copied. If we wait until M phase, mitosis, then the DNA is just being separated and packaged into daughter cells. No new DNA synthesis is happening here. And G1, that happens before DNA replication even starts. So while the cell is active and growing, it's not yet copying DNA. So the latest possible phase where that radio-labeled deoxyadenine could still be incorporated into DNA is the S phase. The correct answer for 4 is D. Let's move on. Problem 5. This says certain ovarian tumors called granulosa cell tumors are known to produce excessive levels of estrogen. A physician who diagnoses a granulosa cell tumor should look for a secondary cancer in which of the following parts of the reproductive tract. Here we want to think about what part of the female reproductive tract is most responsive to estrogen. Estrogen's primary target is the endometrium. This is the inner lining of the uterus. During the menstrual cycle, estrogen stimulates the endometrium to proliferate and thicken so that it's prepared for possible implantation. If there's too much estrogen, then the endometrium may grow excessively, and that can cause an increase in the potential for things like endometrial cancer. The other answer choices, fallopian tubes, cervix, and vagina. These are parts of the reproductive tract, but they are less directly stimulated by estrogen compared to the endometrium. That makes them less likely to show secondary cancer related to estrogen overproduction. So the correct answer for 5 here is going to be C. Problem 6 says, upon ovulation, the oocyte is released into the gland. Now, a lot of students assume it goes straight into the fallopian tube, but that's not quite right. In reality, when ovulation occurs, the oocyte is first released into the abdominal cavity, which is the open space surrounding the reproductive organs. From there, the fimbriae of the fallopian tube sweep the oocyte in and guide it towards the site of fertilization. So even though fertilization usually happens in the fallopian tube, the most accurate answer for where the oocyte is first released is C, the abdominal cavity. Problem 7 says cancer cells are cells in which mitosis occurs continuously without regard to quality or quantity of the cells produced. For this reason, most chemotherapies attack rapidly dividing cells. At which point or points in the cell cycle would chemotherapy effectively prevent cancer cell division? Statement 1 says the S state, 2 says prophase, and 3 says metaphase. So cancer cells, they divide rapidly. So chemotherapy targets phases where division is actively happening or being prepared. So let's break this down. For the S stage, this is when DNA replication occurs. If you block this phase, cells can't duplicate their DNA, so division can't proceed. Prophase, this is the start of mitosis, where chromosomes condense and the spindle begins to form. Many drugs target this to prevent mitotic end. Then there's metaphase. This is when chromosomes align at the equator, and drugs that freeze the spindles are able to halt cells in metaphase. So all three stages are valid targets for chemotherapy. The correct answer here is going to be D. Statements 1, 2, and 3 are all true. Problem 8 says, which of the following incorrectly pairs a structure of the male
Not gonna lie, what the heck?
reproductive system with a feature of the structure? Let's go ahead and work through the answer. Choices. A says seminal vesicles produce alkaline fructose containing secretions. That's correct. The seminal vesicles contribute to the majority of the semen volume, and their fluid contains fructose to nourish sperm, and it's alkaline to help neutralize vaginal acidity. So this is a true association. C says the vas deferens is a tube that connects the epididymis to the ejaculatory duct. This is also accurate. It carries mature sperm from the epididymis during ejaculation. Then choice D says Cowper's gland. These produce a fluid to clear traces of urine in the urethra. This is also true. These produce a pre-ejaculate fluid that clears out acidic urine from the urethra, and also it lubricates the urethra. So this is another correct pairing. Now let's read B. B says the prostate gland is surrounded by muscle to raise and lower the testes. That is incorrect. The muscle that's responsible for raising and lowering the testes is the cremaster muscle, and this is part of the spermatic cord. It's not directly related to the prostate gland. The prostate gland's role is to secrete a slightly alkaline fluid that helps activate sperm and enhance motility. So the correct answer here is B, because that function is wrongly paired with the prostate gland. Now we can move into problem 9, which states, what is the last point in the meiotic cycle in which the cell has a diploid number of chromosomes? Remember, diploid means that the cell has both homologous chromosomes, one from each parent. That separation happens during anaphase 1, and by the time we finish telophase 1, the cell is dividing into two haploid cells. Each one now has just one chromosome from each homologous pair. So the very last point where the full diploid set is still present is during telophase 1, before the cell finishes dividing. That means that the correct answer here is B. Problem 10 says, which of the following does not likely contribute to genetic variability? Let's go through each of these options. A says random fertilization of an egg by a sperm. Since any sperm can fertilize any egg, this introduces a large amount of genetic variation. So this does contribute to variability. B says random segregation of homologous chromosomes. This happens during meiosis 1, and it ensures that the combination of maternal and paternal chromosomes in each gamete is different. This also contributes to genetic variation. C says crossing over between homologous chromosomes during meiosis. This process creates new combinations of alleles, and it is a major source of genetic variability. Then choice D says replication of the DNA during S stage. This step simply makes an exact copy of the DNA. It does not shuffle or introduce variation. It is just copying the genome in preparation for division. So the correct answer here is going to be D. DNA replication ensures genetic consistency, not variability. Problem 11 says, which of the following statements correctly identifies a key difference between mitosis and meiosis? A says in metaphase of mitosis, replicated chromosomes line up.
Oh my god, that's why I hated this chapter, dude. It's always long. Oh my goodness.
In single file. In metaphase 2 of meiosis, replicated chromosomes line up on opposite sides of the metaphase plate. This is incorrect because in metaphase 1 of meiosis, homologous chromosomes line up side by side, while in mitosis, individual replicated chromosomes line up in a single file. So this is incorrect. B says during anaphase of mitosis, homologous chromosomes separate. During anaphase of meiosis 1, sister chromatids separate. This is also incorrect. During anaphase of mitosis, sister chromatids separate. During anaphase 1 of meiosis, homologous chromosomes separate. So this choice gets it backward. So it's incorrect. C says at the end of telophase of mitosis, the daughter.
Ooh, okay, I'm gonna stop lying to myself because my brain is just not working. I have 1 minute and 47 seconds before everything is closed up. Like, oh, I'm going to pass out. So basically, it's been about 2 hours. My brain is about to explode. I feel like I need to walk or do something else. I'm gonna be stretching a little bit and I'm starving as well. Oh my goodness. Oh my goodness. 4 more hours though. 4 more hours. It already passes so fast. I'm gonna keep going for sure. I do have my friend asking me if she can drop me my stuff off. Basically, I was helping her moving in, and um I gave her my vacuum, my ladder, and the brush to wash the sink and everything. And now she wants to bring it back, which is great because I do need to vacuum the apartment. Oh my goodness. So I'm gonna let her know that. I'll be back, you guys. I just need to take a break. I'll be walking around and probably be ordering some food because right now I don't even have the envy to cook anything right now. Not gonna lie. But I'm leaving this here. If I'm going back on live, y'all know the deal. But we've been there for about 2 hours. That's crazy. That's crazy. I wish I had way more patience because I feel like it could have been better. Your school content reminds me of my high school biology class. I almost died. No, for real. I just wanna. Oh my goodness. It's only because I'm alive that I'm trying, to keep it together. Because if it is just me, I'll be like, oh my god, just cussing all along. Like, what the heck is that? I don't even want to be there. Oh my goodness. That's why I hated the beginning of biology. Oh, I hated it. It was just so much content at once. And even though it is related to us, like that's literally how the reason why we're here. But I didn't want to, like, it was too much. It's too much. Like the information is just too much, you know? And now that at my old age, by that time, I'm supposed to be mastering it. But I'm literally trying to review it because my brain is just not. Oh my goodness. Like it's a pain. It's a pain. I'm gonna be honest with you guys. Oh my goodness. It's a pain. Mm-mm. This is just shit. No, my goodness. Okay. I'm gonna be right back. I might take a 1 hour 30 or maybe 2 hours break. I really want to go to bed by 9, hopefully. 4, 5, 6, 7, 8. Yeah. Hopefully by 9, and we'll do the same thing again tomorrow. We'll see how it goes. But now, but I'm just, oh my goodness, it's painful. It's seriously painful. Thank you for the ones who spent some time with me for these last 2 hours. We'll see how it goes. So see you guys in about a couple of hours. Okay.